This video is about the characteristics of life and scientific thinking. So let's examine what is it that lets us know that something is alive. Most of us can recognize this is living, this is not living. But have you ever asked yourself the question, how do I know something is alive? One of the ways that we know something is alive is that they have similar characteristics. And often we use these characteristics to determine, is something alive? So what are some of these characteristics? Here's a picture of an ultrasound. Let's take a look at some of the things that all living things share. For example, all living things are made of cells. So a living thing has to be at least a cell in order to be alive. Living things grow and reproduce. Growth just means an increase in size, and many living things, that's what they do. Often, living things also develop, that is, they go through life stages. Living things reproduce, so whether that's asexually, like a bacteria, or sexually, where there's passing on of traits from parent to offspring, living things grow and reproduce. Living things also have some kind of chemical reactions. Often those are related to energy. We call all those chemical reactions, especially those related to energy, metabolism. Homeostasis just means maintaining the internal environment. So your heart rate, your blood oxygen level, your temperature, your body is going to work to maintain that internal environment. So you might sweat if you are hot. You might shiver if you are cold. That's your body maintaining homeostasis. Living things contain DNA. They contain all kinds of organic molecules, proteins, carbohydrates, lipids, but DNA is a unique molecule to living things. DNA stores and transmits information. Living things respond to stimuli. That is, they can take in information from the environment and then they react accordingly. Living things evolve and adapt. So they're changing, or they have features that help them to do better in certain environments. So if you take a look at this ultrasound, we can make inferences and observations about this baby and how they have some of these traits. For example, we could take an amniocentesis, some of the amniotic fluid, and we could discover that the baby is made of cells, that there's DNA, half of that DNA comes from the mother, so there's going to be some similarities with the mother, some similarities with the father. We can observe the heart beating and recognize that the baby's maintaining homeostasis. We can see the umbilical cord, how the baby's getting nutrition, and those chemical reactions are going to allow the baby to do things like move, for example. We can see how the baby responds to stimuli as the ultrasound wand presses against the mother's abdomen, and the baby might wiggle and squirm as a result. We can infer that the baby is growing. We can infer that reproduction happened because of the existence of the baby. The baby didn't start out that size. The baby increased in size, and so that's evidence of growth. So we can see these characteristics of life. Some we can observe, some we need to infer. Living things are also organized by levels. Bigger things are made up of smaller things. Atoms make up everything, but actually atoms are made up of things, protons, neutrons, and electrons. But atoms make up molecules. As atoms join together, they make up complex molecules. Molecules in turn make up organelles. Organelles are just the parts of a cell. Organelles make up cells, and there's all kinds of different types of cells. But when you put similar cells together, they make up tissues. So for example, muscle cells make up muscle tissue. Tissues then make up organs, like your heart is made of muscle tissue, but it's also made of nervous tissue, and it's made up of connective tissue. Organs make up systems, like the cardiovascular system, which is your heart, which is an organ, but so are your veins and arteries. Systems then make up an organism, and in this class we're going to study 11 body systems that make up the human organism. Organisms together make up a population. So you might say all of the cats in a town. That would be the population of cats. All of the deer in a certain forest. That would be the population of deer. Once you start adding different populations together, that makes a community. And then when you put different communities together and you add the abiotic parts, the dirt, the sunlight, the temperature, the rainfall, that makes an ecosystem. Large ecosystems together make up a biome, and then biomes together make up the biosphere. So biomes are things like deciduous forest or desert, tundra, things like that. Biosphere is this little area around the Earth where life exists, just on the surface of the Earth. Let's say you were to see an advertisement that looks something like this. When it comes to scientific thinking and reasoning and the scientific method, we want to apply those critical thinking tools to things that we see like this, because we want to check to see, is this claim valid? This product claims that the Wonder nutrient can help you lose weight. We want to know, is that claim valid? So we might ask some follow-up questions, like, is this type of weight loss consistent with other research or sources? Is this source of information reliable? The person or organization making the claim, are they a reliable source? When they were testing this claim, did they use proper scientific procedures? And when they did they interpret the results correctly? When they looked at those results, did they read that graph or read that data correctly? And are there any other possible explanations? Was it really the Wonder drug that was making the difference? Or was it an increase in exercise? Or was it more conscientious eating? Like, what was it that really caused the weight loss? So when we're looking at something like this, we want to evaluate it critically. When we're going through the scientific method, we use certain steps just to systematically approach problem solving. Now, we do this all the time for all kinds of things. And we often don't think about the steps, but these are kind of the steps that we're taking when we're making a systematic approach to solving a problem. Observations are based on the five senses, things that we can see, hear, smell, taste, or touch. So we make some observations about the world around us. Our hypothesis is based on deductive reasoning. The way deductive reasoning works is that we come up with what we think is a conclusion. All right, so that's our hypothesis, an explanation. Then we look for facts to support that conclusion. So we test it, we accumulate data, and we see if that data supports the conclusion. If the data does not support the conclusion, then we throw it out. So think of it kind of like an upside down funnel. At the top of the funnel here, we have our hypothesis, and then it's supported by the facts that we discover. Inductive reasoning is the opposite. We look at the facts, and from the facts and data that we uncover, we form a conclusion. The way science works is we come up with a hypothesis, and then we see if the evidence supports it. Deductive reasoning. When we design an experiment, we want to make sure that we include a control group. A control group lets us know if the variable that we changed actually had an impact, actually changed anything. So we have a control group. We might give them a placebo. A placebo is something like the experimental group would get, but it would be like a sugar pill or a water tablet. Then we're going to try to control as many variables as possible so we can test one variable at a time. That's not always possible, but a good experiment is going to eliminate as many variables as possible. The variables that we manipulate or control are called the independent variable. So I control it independent. The dependent variable is the thing that's changing, and we want to observe those changes and discover those changes. That's the dependent variable. Once we see those changes, we're going to analyze those things. We might put that data into a graph or chart to help us see the trends, and we're going to analyze that information. What is it telling us? Finally, based on all that, we make a conclusion. So let's go back to our claim that this Wonder nutrient is going to melt away huge amounts of weight. And let's see. So we make this observation, this nutrient might affect weight loss. We come up with a hypothesis. If the Wonder drug is related to weight loss, then the more Wonder drug, the more weight loss. So how do we set up that experiment? We get a control group, give them a placebo. Then we get group 1, and we give them a small amount of wonder drug, 1 gram. A group 2, we give them a larger amount of wonder drug, 10 grams. Group 3, we give them 100 grams. Over the course of the next 2 weeks, we try to eliminate as many variables as possible. Everybody eats the same thing. Everybody sleeps the same amount. Everybody exercises the same amount of time. All those variables stay the same. The only thing that is really different about their course of life is that some are taking this wonder nutrient, some are not. Some are taking it in greater quantities than others. At the end of our 2 weeks, we compare the results. What's the average weight loss of the placebo group? What's the average weight loss of the 1 gram group, of the 10 gram group, of the 100 gram group? And we begin to see if there's any kind of trend, and if that trend is connected to this wonder drug. So we take that data, we analyze it, maybe in a bar graph, maybe in a line graph, but we take that data and we analyze it, and then we kind of see, like, what was the result. Whenever we're doing an experiment that especially involves humans, we first want to make some ethical decisions, like, will this be actually helpful to humans? And we might start those experiments on animals to see if there's any adverse side effects. After we do some animal testing, we'll first test on a small population, and then on a large population. Those tests will be voluntary. Based on that information, we come to some conclusion. Was this productive or not? Was this new drug or weight loss thing better than what we already have on the market? Now, sometimes these experiments can be influenced by bias. For example, if I did the experiment on my own drug, that would be biased because I want it to be successful. So I might tweak the data to make it look better than it is so that I make lots of money. That's experimental bias. Other things might just be experimental error. For example, I know the placebo group and I know the experimental groups. A double-blind experiment means that nobody knows which group they're in. I don't know which group they're in. That way, it helps to alleviate the bias of the experiment. I hope those things are helpful as an overview of the scientific method and the characteristics of life.